for that. So welcome everybody to our final Landcare Builds Skills for 2024. We still have 2 more Landcare checks in. And today we're going to be learning some skills around soil management practices. And I'm going to be honest with everyone. This is a subject I know embarrassingly little about, but it also fascinates me. And I have selfishly been looking forward to this event for a few months. I think we can all agree that soil health is critical for our planet and for all life that it sustains. And if we can pick up some practical knowledge and skills about getting the best from our soils, that's a win for everyone. So before we get started, I'd like to acknowledge that I live, work and play on Wadandi Noongar Boodja, and I pay my respects to elders past and present. I extend that respect to any Aboriginal or Torres Strait Islanders on the call today and acknowledge their connection to land, sea and sky. Please feel free to acknowledge what country you're joining us from today in the chat function. I'd love to know. I'd also like to acknowledge our funders who I believe will be dropping in today to say hi. So thank you to the WA State NRM program and also the National Landcare Network for supporting WALN to deliver these skills building opportunities to our members and the broader community. I'm going to introduce our first guest speaker. We're going to try and keep questions till the end if that's okay. Unless you have a burning question, we'll wait for both Alan and Mark to finish presenting. So our first speaker today is Alan Walker. Alan has studied the soil food web and soil microscopy, completed a graduate certificate in regenerative agriculture at Southern Cross University, a permaculture design certificate at Fair Harvest, and holistic management with inside-outside management. She's keen to share her knowledge and information and teach practices that will assist in developing and nurturing healthy, nutrient-rich soil. Ellen is the founding partner of Earthwhile Australia. Ellen, I'm going to hand the floor over to you.
Thanks so much, Casey. And I'm coming to you from Noongar Boodja land. So I'd like to acknowledge our respective elders, past, present and future. So this is quite an exciting time to be able to share our learnings with you, and I'm going to get pretty much right into things so that I know that we have limited time. So let's hope that we can get this right from the beginning. So hopefully you can see my screen, and excellent, thank you. So our work is very much premised on a quote from Sir David Attenborough, who says that no one will protect what they don't care about, and no one will care about things that they've never experienced. And so our soil microbes are integral to life on Earth, and yet so many of us have not had the opportunity to view our own soil microbes. And even though we can't take you right there today, hopefully we're going to get you better acquainted with soil microbes, with their role in our soil, with if you've got a microscope available to you that you might be able to then go on and identify them when you're looking at some soil samples and so on. So let's start with thinking about soil health. And if we think about soil health, it's actually going to change depending on what it is that we're planning to grow. But there are 3 main aspects: the physical aspect of the soil health, that's the structure and so on of it; the chemical aspect, so the pH and the minerals and nutrients that it contains; the biological aspect, which we are hearing more about, but has been pretty much put in the background for a long time. And that's about not only the earthworms and insects and macroarthropods, it's actually about this tiniest little microbes that we can't see with our naked eye: the bacteria, the fungi, the protozoa and the nematodes. So, and also some people will throw into that mix the energetics, but today our focus is going to be on biology. The reason I wanted to show you this particular slide is because even though we're going to be talking about biology today, we need to remember that it sits within the physical aspects of the soil and those chemical aspects of the soil. They all work together. So biology has been basically put to the back burner because we've had so much focus on the chemical nature of soil that if we add this, it will make things grow and so on. So biology, the biological part of things, it's not new, it's been around. So some of you will know David McFall. David's grandfather had an article published in the Farmers Gazette back in 1929 on soil microbiology. So not new, but just being foreground again. So what is this stuff? So if we think about in a teaspoon of healthy soil, we can expect to find more microbes than people living on this earth. Can you imagine that? So that's billions of bacteria, kilometers of fungi, tens of thousands of protozoa, and hundreds of nematodes. And I wonder in amongst you whether some of you are cringing at that word of nematodes or not. But anyway, let's see. They are all amazing creatures. And we know that when we have all of these organisms in the soil from the biological aspect of things, that we've got something that's quite healthy. So what do these things look like? Well, this is a quick introduction before we get into things a little bit in a bit more depth. Basically, in here, we've got our bacteria. So the bacteria are these little bits and pieces here, these little tiny roundy bits here. Fungi is these long strands, or some of them can be short, but basically you can see that they're quite different from the bacteria. Protozoa can come in different forms. This particular one is a little amoeba, but you can see again, this egg-shaped amoeba, which we can fairly easily identify, is quite different than the fungi or the bacteria. And there we have our good old nematodes, which might be hiding behind some of your pictures, but the nematodes look a bit like a worm, but we'll see that they move a little bit differently. So these are our 4 microbes that we're going to be talking about. So why are they important? Well, they build soil structure. The structure is soil structure is basically integral to moisture holding and to cycling nutrients. Would you believe that soil microbes play an important role for plants to be able to resist pest and disease? And they also help detoxify our soil. So if we've had things on there that we would rather that we didn't have, then they can actually break down those toxic substances into their elements, their carbon, their hydrogen, their nitrogen, so on. So that's in general terms. We're going to talk about those things a little bit more through the presentation. But how will we identify these microbes? Well, just as you can see the bee here, and I bet you can see the flower and you can see the leaf, it's quite easy, isn't it? And it's because those things have different shapes, they have different sizes, and they have different movement. And that's how we're going to identify the microbes. You might be thinking, well, hang on a tick. Is this a bit nerdish? Is this something we want to do? I suspect that many of you, particularly those of you in the Landcare movement, are going to have a microscope tucked away in the back room. And maybe it hasn't seen the light of day for a while. Well, hopefully you'll bring it out after this, because when you can identify the microbes and if you can see them through the microscope, you know that you've got them in your soil. So this is what we're going to be trying to achieve today. So here we have our bacteria, and the bacteria we know from before when I showed you, it's these little dots here. But when we actually set them moving, you can see how they move. So bacteria, it's about them being small. They have these different little shapes, but it's that jiggling sort of movement. I wonder if I can do that one again. I might just have to go back to there and here, just so you can check that out, all that little movement, which is pretty cool. So bacteria bind together the smallest of the soil particles, the silts and the clays, and make them into what we can think of as house bricks. House bricks by themselves aren't necessarily much use, but if you think about it, a house brick isn't likely to blow away or wash away like the particles of soil will. So they're very useful there. Bacteria also gather nutrients. So they are involved in breaking down the rock and the stone, but have a huge role in decaying organic matter in our soil. And when they do that, they're basically putting up this ooze, and through this two-way thing, they then suck back in the nutrients, but they hold those nutrients in their body. Lowenfels and Lewis suggest that it's a bit like a bag of fertilizer. So the nutrients are coming into their body, but they're not available to plants like that. So you can think, well, okay, but what happens when they keep eating? Well, that's pretty amazing, because when they keep eating until they get double in size effectively, and they actually split in two. So you end up with two. So bacteria can reproduce in something like 20 minutes in good conditions. Bacteria prefer foods that are high in nitrogen. So that's useful to remember for when we might want to have more bacteria. And I expect Mark's probably might talk a little bit more about some of that aspect of things in his presentation too. So moving on, the next one is fungi. So we've got a couple of main functional types of fungi. There's the mycorrhizal fungi, which has an association with living roots. And there's and mycorrhizal fungi, we can see through the microscope, but we can't actually identify whether it's a mycorrhizal fungi or whether it is a root hair. So we won't be actually looking at those when we're looking through the scope today. The other form is the saprotrophic fungi, and the saprotrophic fungi is like this one here, and it's the one that does a lot of it, or does the decaying in the soil. So fungi is particularly useful, again, in structuring soil, because the fungi put out these long, sticky strands, which you might think of as being like mortar. So we've already got the bricks that the bacteria have formed, and now we've got mortar. So you can just imagine, hopefully, that with bricks and mortar, we could create these amazing structures in the soil. So with big rooms where there's lots of air and water could actually flow through, where roots can get in, and little rooms where there may not be much air, where those little tiny bacteria can hide from predators, and where some of the microbes actually don't need much air, and so they can operate as well. So fungi have a role in that part for building that soil structure, but also, like bacteria, they gather nutrients, take nutrients into their bodies, but they don't release them to plants. So they're another bag of fertilizer. So I want to talk a bit more about mycorrhizal fungi here because of how important they are. So they have an association with the living roots of plants. And so even though this diagram here is showing trees, for those of you who might be in crop growing areas, this applies to crops as well. So basically, there's about 90% of plants have or form this association with mycorrhizal fungi that then effectively extends the roots to up to like 1,000 times the area that they can, and the roots could cover. And they take in water and they gather particularly things like phosphorus and calcium and nitrogen. Zinc, I think, is another one. And so they are just really, really important. They are something that we have seen decimated to a degree over in the wheat belt country, where we have seen a bit of excessive use of synthetic fertilizers and chemicals. So they're particularly sensitive to those things. So I'll leave that there. And if you've got some more questions about those, come back to me as when we get to the question time. What we are going to be seeing under the scope today is the saprotrophic fungi that play very similar roles to the mycorrhizal. And in fact, in some plants, so I mentioned that most plants have this mycorrhizal association. Some, like the brassicas, don't. And in those, you can actually see these little saprotrophic type of fungi inside the plant, would you believe it? Inside those brassicas. Anyway, the saprotrophic fungi, we can tell that they're fungi by their shape. They're long, or they're longer, they're like twig shape. They have uniform diameter. If you can see the ends that have flat ends, and sometimes they have these little septa. Can you see these little septa here that are a bit like bamboo segments? So they'll have those things. And so hopefully you can tell the difference between our fungi and our bacteria. So saprotrophic fungi play a big role in decomposing, in that they contribute to the cycling of nutrients, those bags of fertilizer. we talked about that building of soil structure, those sticky strands that are actually going to create that underground structure. And they prefer foods that are high in carbon. So think wood chips, cellulose, lignin, hydrolysates, fish hydrolysates, that type of things. So if we're thinking about soil structure now, in real life, we would normally do this one, as I say, practically. So I'm going to struggle with this because this presentation is, hopefully you can imagine it, but I feel strange just talking to a slide with it. But effectively, if you think about this is flour. So basically we've got half a cup of flour here, and flour is effectively like dirt. It might have a few bacteria in it, but basically it doesn't have much in the way of living organisms in it. Then if we think about a slice of white bread, so that's the white flour, and we're adding the organisms in the way of yeast or fungi in the form of yeast, and there's probably a little bit of sugar, so we're getting a few bacteria as well. So when we have that, and then we have some rain, so the next bit is going to be that it's going to rain. And so here, oh, what's happened to my rain one? So this is going to be, we would pour some water over that, and just a small amount over the flour, we'll see that it just runs off and pools around. When we pour it onto the bread, we can keep pouring. It's actually quite surprising how much will go in there, and it's all just soaked up. And that's because when we have soil structure, we've got effectively a soil sponge, what an Australian microbiologist, Walter refers to as the carbon sponge. So carbon can hold so much more water than, carbon can hold something like about 8 grams of water for every gram of that it is. And would you believe that microbes are predominantly carbon? So we're going to see that soil sponge effect. So just imagine then when you start to dig into this, into here, and when we dig in there, if that was a plant in there, we'll find that it's just dry. So, and you're just going down into dry flour, dry dirt. When you dig into here, we find that we've got like this sponge effect. And so that plants can effectively manage the soil, the moisture in the soil. So it means that the soil is going to stay damper for longer. And of course, microbes are like us. They actually need to have moisture to be able to survive. So that's really useful. So in this one, it's, I think Gabe Brown said, it's not how much rain or water you put on, it's how much you can keep in the soil. So we often think about building dams and doing other things, which all have a role, contour banks and so on, they all have a role. But just imagine how much extra water your soil could keep if it was like a sponge. So the fungal to bacterial ratio, this fungi and bacteria, that these two microbes that we've talked about so far, also play a role in what will grow. So research from around the world has revealed that basically weeds, or when we're talking about weeds here, talking about simple annual plants, weeds grow in a bacterially dominant soil. When we talk about fungal to bacterial ratios too, we're talking about the biomass, not X number of fungi to the X number of bacteria, but the weight or the biomass. And so as we're moving through, you'll see that as we get more fungi in the soil, then we might be able to grow our grasses and then basically our forbs. And for those of you growing row crops, you'll get up to being able to sustain those deciduous crops, the orchards and so on, and up into our old growth forests where we get very quite highly dominant soil. So the relevance of this is if you've got annual weeds growing, it's likely that you've got a very bacterially dominant soil. So let's look at how you might rebuild that fungi in the soil to be able to get up to these higher levels, higher complexities of plants. So remember that we had those bags of fertilizer, the bacteria and the fungi. So now we need to think about how we're going to get those out to the plants. They're great as bags of fertilizer in the shed, but not much use to the plants. Enter the protozoa. The protozoa work as grazers and spreaders by eating the bacteria, and particularly protozoa, particularly like bacteria, keeping in the nutrients that they want for their own development and excreting the rest. I'll tell you a little bit more about that in a moment, but let's just have a look. So in here, we've got an amoeba. So there's three varieties, I suppose, of protozoa. Amoeba, which this one's a testate amoeba. This one here, this egg-shaped stuff, white stuff here, light stuff here is a naked amoeba. And then we've got ciliates and flagellates. Now, ciliate, in this next little bit of video, you'll see is the big one, and the flagellates are the microbes that are zooming around a little bit more. And so there you go. Can you see that big hairy one? So that's a ciliate. And then these other bits that were there and there, like this one here, when they were moving, they're flagellates. So they obviously don't stay still long enough for us to take a nice photo of them and just sit there and smile. But they're actually working in our soil and they're releasing these nutrients. They're the grazers and spreaders. A little bit of chemistry. If you consider that a bacteria has a carbon to nitrogen ratio around about 5 to 1, so 5 units of carbon, 1 unit of nitrogen. Protozoa are said to have a carbon to nitrogen ratio around about 20 to 30 to 1, let's say 20 to 1. So protozoa come along and eat 1, 2, 3, 4 bacteria. They've now got 4 times 5 units of carbon, tick, that's 20, so that tick that. But they've also got 1, 2, 3, 4 units of nitrogen, and they only needed 3. Oh, sorry, they only needed 1. Gosh, my maths is going. So they excrete those other 3. And that becomes in a plant available form that the plants can take up. So they can access this nitrogen from within the soil. But it's got steak knives. It's because it's not just the nitrogen that it gets excreted in that process. It's the hormones and enzymes, which if you're into food, you might know those as phytonutrients. And you will perhaps know that that's what actually builds our immunity. So it builds the plant's immunity to pests and disease. How awesome is that? So all these things, it's all about microbes. Let's get into our nematodes. So our nematodes, whereas the protozoa had this carbon to nitrogen ratio of around about 20 to 1, nematodes are said to have a carbon to nitrogen ratio of around about 100 to 1. So just imagine how much processing they can do. And it's not just carbon to nitrogen, it's carbon to phosphorus, carbon to sulfur, carbon to boron, carbon to some obscure germanium or something along those lines that plants might need at some stage. So our nematodes, so hopefully you could quite easily identify that a protozoa is quite different to a fungi or a bacteria. And so moving on to our nematodes, again, they're different again. And so as much as they look like a worm, they move like a snake. So there we go. And so they also are making these little tunnely bits in the soil. And so when we talked about that structure with those spaces where, because let's face it, if a protozoa can eat something like 10,000 bacteria in a day, they're going to need somewhere to hide so that there'll be some to reproduce for tomorrow because of that many. And so the nematodes too can't access the bacteria when they're hiding in some of these little spaces. So we need all of those opportunities in the soil with that soil structure. So some more nematodes here. So let's go down to this one here. So this one here, for those of you who are growing ryegrass, if you come across ryegrass toxicity, this nematode will be may cause some problems or does cause the problems with ryegrass toxicity. And you may also have run into it with your tomatoes, growing your tomatoes or something like that, those sort of crops. But the good news is that they're only about 5% of the variety of species of nematodes. And so we can think of them as doing a couple of good jobs. One, indicating that our soil microbiology is out of balance. And the second one is that our plants aren't healthy. Because if our plants aren't healthy, they can be subjected to disease so much easier. This one here, oopsie daisy, let's go back. We're not quite ready to do our recap. So this one here is a fungal feeder, and it has a spear that shoots into fungi and can suck out the cytoplasm. This one here has a big wide mouth, and it's going to be able to suck up and vacuum up the bacteria. And this one here, if you've got these, you want this one. This one's a predatory nematode. So that's our little range of nematodes. And so just as a quick recap, we've talked about our fungi, we've talked about our protozoa. You're not going to be able to actually see it in there because the movement, we haven't got movement here. And the nematodes moving in there, and our bacteria. So hopefully those things are familiar to you. So I'm going to stop sharing this screen now. Oh, actually, I'll just go to this next slide because I'm going to stop sharing here because I want to show you some microbes in real life. Let's have a look and see. Aha! Can you see what's happening here is Georgina's on the other side of me and she's got the microscope, and so that's coming live. So Georgie, can I have the test tube, please?
It's open, please be careful.
Oh, okay, I won't spill it. So what we've done, so this is just a little bit of compost in here and some water, and Georgie's taken 1 drop of that, and we're now looking at this at what magnification?
400.
Sorry, what? No.
400.
Yeah, so this is 400 times what we'd see with the naked eye. So this is a pretty small nematode. But can you see all of the bacteria in there? So you start to understand this is 1 drop from 1 mil of soil. And so you start to understand why we are seeing like billions of bacteria, why there could be billions of bacteria in this teaspoon of healthy soil. So we've got aggregates in here. We've got those, oh gosh, those little bacteria are going crazy. So and this one, oh yeah, so Georgie's just getting us to see what else we're seeing here. Oh yes, can you recognize that? So that one's a piece of fungi. So we've got fungi in here. We've got the bacteria, and there, oh, shooting off there was a protozoa. There's another protozoa in here. Have we got any testate by any chance? Let's have a look quick. Oh yes, and here down here we have a testate amoeba. So hopefully with those different bits that you can see, that you can pull out the microscopes that you might have available to you and at least see if you can find these little organisms in the samples that you can take. So I know we're a little bit pushed for time. Oh, Georgie's just given me the 5-minute sign. So I'm going to stop sharing from here and let's go back to our presentation one. And so we've looked at some real microbes. It's always fun when you look at your own, but at least hopefully this is a little bit there. So let's think about why these are important. We've talked about soil structure, but now let's talk about the nutrient cycling. So if we think of that plants have, our food, we digest our food in our stomachs. So in there, and our microbiome in our stomachs digests our food. Plants don't have a stomach as such, so they outsource their stomach to the soil. And so the soil is the plant stomach and the microbes do the heavy lifting. So I've got there, don't give your plant a kick in the stomach. So you can imagine that a kick in the stomach is going to upset that soil microbiome, or the soil microbiome, which is going to have impacts for what the plant can do. So when we think about, so trying to cram in half an hour in half an hour, what we could talk about for days. So hopefully you'll get some ideas and ask extra questions later. But when we think about getting food, this is an important part, because when nature feeds the plant, then it's going to use processes like the predatory grazing that we talked about with the protozoa eating the bacteria. There's something called rhizophagy, where the microbe is sucked up through the plant, the root hairs, and the plant actually uses a strong chemical, basically takes off the cell walls of the microbe, gets all the nutrients and spits out the DNA. And would you believe that these microbes then, many of them still survive and keep going? There's the mycorrhizal fungi, where it has that association directly with the living root and it swaps carbon for the nutrients. So all of these things mean that our plants end up with enhanced resilience. They all require microbes to do that. When we start with humans feeding plants, we tend to feed the plants what we think they want when we think they want it. We'll do that to the best of our ability, but we're still in control, which can develop a potential dependence on humans. With that dependence on humans means that if we're a bit out of kilter or something, or weather situations change, or there's predators come in that we haven't allowed for, then the plant is going to suffer. Whereas when the plants can access all of these feeding arrangements by themselves, they can just reach out with control, that I want an extra blanket to stop that frosty night. I want some antibiotics because I need to have, I'm not feeling too good. I've got predators coming. I'm going to put out the the anti-predator thing. And so they are so much more resilient. The plant health pyramid, and this is something that you can access online quite easily, is important because it actually ties together this notion that of when we looked at those circles of the chemical, the physical, the biological, this ties together particularly the biological and the chemical. So in the bottom couple of layers here, that's dependent on having a balanced range of minerals and nutrients in there. As we move up, so by the time you get to this top layer, you're not going to get the brassica or white flies on your brassica, so maybe on your canola. In the top 2 layers, that's dependent on active, diverse microbiology. If you think about active, diverse microbiology, what does that mean? If you think of microbes as being a little bit like humans in the sense that they have all these different talents and skills, I couldn't build a house by myself this weekend. But if I had enough architects, bricklayers, plumbers, electricians, urban planners, tea makers, definitely some conflict resolutionists, logisticians, it could happen. And so this is where, when we get all of these microbes from a variety of places, then we're going to have very nice, healthy soil. How do we know if our soil microbiology is healthy? So for out there, so basically, if you pull up a little tiny seedling and you have it, or the roots of your plant, provided it's not a brassica, even though brassicas will still have this to some degree, you'll find that the plant has been putting out exudates. So we haven't even covered the exudate side of things, which is really important because that's what feeds the microbes. But it'll be putting out these exudates, that that sticky substance effectively that the microbes produce is going to hold that soil to the plant roots. So these bare ones here, not much happening here, but on these ones here where we're getting these like dreadlocks, that's good. You could use a piece of calico, and so unbleached calico, bury it in the soil. Usually you do this, and you can use the cotton undies. You might have heard of the cotton undies test. So you can do this in autumn and spring, leave it there for about six weeks or so, and then mark where you put it and then pull it out and see if it's pretty much decomposed. Then it's likely you've got quite a good range of microbes. The refractometer, this isn't the whole picture, but hopefully you might have heard of a refractometer. A refractometer measures total dissolved solids, but it has an association with the rate of photosynthesis, and we're ultimately wanting to optimize photosynthesis that is also reflecting a good microbiome. And then, of course, you've got looking through the microscope, which we've been able to do quickly here today. So for happy soil microbes, they will thrive with the minimal and thoughtful use of fertilizers, tillage, and science. We're not fundamentalists. It's not about not doing things, but it's being about thoughtful about what is going to encourage the life of your microbes as opposed to what's going to kill them. Having living plants all year round is incredibly important because living plants put out exudates in the soil to feed the microbiome, and that the diversity of plants, oops, sorry, just so Georgie gave me the roundup time. The diversity of plant families, each plant family has its own microbiome, and so it's very important to have that diversity of plant families. So recapping, microbes build soil structure, they hold moisture, they cycle nutrients, they help with disease and pest resistance, and they break down, or they detoxify things. So thank you all so much, and I hope that you'll have a greater appreciation of your soil microbes. No one will protect what they don't care about, and no one will care what they've never experienced. I really hope that you get the opportunity to experience your own soil microbes and care and protect them. They are integral to our life on Earth. Thank you.
Thank you so much, Ellen. That was so awesome. Your very real passion was coming through there in your storytelling. So thank you so much for sharing all of your knowledge. Microbes are obviously superheroes, so thank you for pushing that point forward. If anyone's interested in seeing graphics from a mycorrhizal fungi scene, Bob Brown's The Giants documentary has the most stunning little piece on mycorrhizal connections, if anyone was interested in having a look at that. I am going to hand over to Mark. He is our second speaker today. Mark has been actively involved in the fields of organic and biodynamic production, permaculture, sustainability, agroecology, and holistic management for the last 30 years. He's worked on a number of educational, research, community and commercial projects throughout Western Australia, and in between times managing orchards, livestock and market garden enterprises on various properties. He is a content creator for the Lower Blackwood LCDC, which some of us might also know, and he is the Witchcliffe Eco Village's horticulture manager, which is not too far from where I live. So take it away, Mark.
Thank you. Okay, we better keep rolling straight into it because we've got time is of the essence. So I'm just going to kind of reiterate a couple of things that Ellen talked about, but just follow it on more with management practices and, you know, how we can go about sort of encouraging or building these populations of microbes, beneficial microbes in our systems. So whether it's in the home garden or on the farm or an orchard or whatever it might be. And so to do that, we just got to understand a little bit about the different microbe groups and what, you know, what their needs are and how we can actively cultivate them or culture them. Just straight off the bat, I just wanted to show you this. There was a study done, you know, a decade or so ago on all the living things on the planet, and you know, we as farmers, we are growing living things, and so it helps us to understand a bit about the system, that, you know, the blueprint for life on Earth. And when we look at this, there was about, in the audit, they sort of ascertained there was about 550 gigatons of living things on the planet, of living matter on the planet, and of that, 450 gigatons was the plant material, which is the big green the plot there. And so the vast majority of life on Earth is plant material, and that's for good reason. I mean, the plants are the ones that are photosynthesizing and basically creating carbohydrates, which all the rest of us living things rely on as a source of energy, but also they're the building blocks of organic matter. So yeah, needless to say, plants are really important organisms in our systems. But then you look at the next biggest groups. We've got bacteria at 70 gigatons, fungi at 12. We've got archaea and protists, some of these things that Ellen was talking about. Yeah, these microorganisms that you can't see with the naked eye are still, you know, you can't see them, but they're still making up a large portion of the life on Earth. And they're obviously very essential for all those processes that Ellen talked about. And all of that is needed just to support this measly 2 gigatons of animals, which is us and the animals that we farm. And so unless, you know, and even then, when you break down the animals, the largest groups, the insects, there's a large group of fish too. So we're looking at nematodes, mollusks. So humans and domestic animals really is a very small fraction of life on Earth, even though we're big and important. So we really need all these other networks in this biology, these organisms that organize things. They organize organic materials that really become our food and our sustenance, and they carry out all these different functions, even go so far as sort of helping us with climate regulation and things like that as well. So yeah, needless to say, they're all really important, but just reiterating that plants are where it all starts, creating that food source for everything. And then there's a whole lot of other things that happen after that. Basically, the microbes that live on and around plants perform all these services, some of which Ellen talked about. And, you know, they do this in exchange for their energy source, their carbohydrates from the plants. And normally the things that they do are beneficial to plant growth because it's in their best interest to support the growth of plants if the plants are going to supply them with all their food. So they do wonderful things like fix nitrogen from the atmosphere, make it available to the plants, acquire minerals from the soil. They deliver the minerals to the plants. They cycle elements, so like in organic materials, they break them down and release the nutrients so the plants can use them. They assemble all these useful compounds that are, you know, help plants grow. They provide protection or, you know, increased immunity against diseases, promote growth. They also produce this humus, which is the sticky stuff that binds soil particles together and lasts for, you know, longer periods of time in the soil, and that's what creates a crumbly, structured soil that allows air and water to penetrate and disperse. And, you know, they modify the conditions around the roots, so pH and oxidation states and all these things just to help plants grow better so that plants can keep feeding them. They're the meal train. And, you know, they move around and distribute things and everything. So there's a whole network of stuff going on under the soil and even above the soil, which involves these invisible organisms. I just wanted to break the sort of biological players into these different groups, because it helps us to understand how to manage them if we understand a bit about their requirements. And so these sort of groups are kind of handy sort of groups to get that understanding. So starting off with the symbionts, which are the things that actually associate with the plants, and they sort of live, you know, in close vicinity or inside the plant, so around the roots, on the leaves, in the seeds, in the flowers, and they're performing these services for the plants in direct exchange for what the carbohydrates the plants are giving them. And then you've got your animals, which includes big animals like cows, but also smaller things like slaters and worms and things like that. And they also perform services, but they're not living on, they're not living directly associated with certain types of plants. Animals are a bit broader. They might be eating a range of different plants, and they move around, and they also eat a range of different sort of, the residues, the organic materials that end up on the soil surface. But then you've also got the decomposers, which primarily eat they're really the proper residue eaters, and that includes animals like slaters and worms, but it also includes a whole lot of microbes, like Ellen showed in our slides, like the fungi and the bacteria and the protozoa and things like that as well. So these different organisms have different food sources. The symbionts need, plant exudates. They live close to the plants and eating the stuff the plants are producing in real time. The animals are eating sort of high value foodstuffs that, plants ultimately, when the plants grow, they eat the actual plant material. And then when the plants die, the decomposers come in and they cycle those nutrients. So just starting on the symbionts, here's just a picture of some root exudation from a root of corn, an aerial root, so above the ground. And it's just showing you the sticky nature and the syrupy, sugary, rich substance that plants are producing to feed microbes that are living on them. And this would be feeding nitrogen-fixing bacteria and various things so that they can provide those things for the plant, the corn. We've also got nitrogen-fixing bacteria that live in the roots and associate with legumes like our rhizobia. And I'm just going to flick through these quite quickly because we've got a lot to cover in half an hour, but I'm sure most of you are familiar with that. And Ellen talked about the mycorrhizal fungi, which, is attached to plant roots and stretches out. It's very microscopic, so you can't really see it very easily unless you've got very powerful microscopes. And, they can get into very good at getting nutrients from all the nooks and crannies in the soil and delivering them to the plants and so on. And again, the rhizophagy, where the plants are actually eating whole bacteria, they mostly do this in the root tips, and then they pretty much, absorb the cellular contents of those bacteria. So they're getting complete meals of, amino acids and enzymes and vitamins and all those things from eating bacteria rather than just purely soluble nutrients, inorganic nutrients. And like I say, this mostly happens in the root tips. So when plants first germinate and a high portion of their roots are root tips, it's really important that they are able to access these bacteria and eat them. They're a very important formative food source for plants. So it helps if they germinate in healthy living soil. Animals, really important. Aside from the big ones, there's also the little ones we talked about. But one thing about animals is they have this digestive system. So they've got mechanical means of breaking things down, whether it's teeth or, grinding or whatever it is, to break materials into smaller bits like straw and things like that. And then, they've got enzymes and acids and stuff in their stomach, as well as a whole population of gut microbes. So they can really break down organic matter very quickly, which then releases the nutrients back to the soil and to the plants to help them grow. So when you think about it, plants are not going to grow very well in a bale of straw because all the nutrients are still tied up in the straw. But you feed that straw to a cow, and then it comes out after the digestion process as manure or urine, and it's rich, and plants can then actually utilize the nutrients that were in that straw in the first place. The animals also have this ability to sort of, chop and prune things and instigate lots of regrowth. They, sometimes keep weeds and populations, pest populations in check. You've got things like your bees and insects that pollinate plants. things like dung beetles that incorporate organic materials into the soil, and the worms and everything. All of those are burrowing around and helping with soil aeration and structure. I've just got a couple of pictures of some insect animals. These on the left, there's some predatory wasps. They've deposited some wasp eggs in a caterpillar, and it's like the hungry caterpillar story. The caterpillar keeps eating while the wasp larvae develops in the caterpillar, and then eventually they emerge and leave a skeleton of a caterpillar with holes in it, at this point dead. And you can see the cabbage leaf on that picture. So it's a cabbage eating caterpillar, and the wasps have basically predated on it, and now they have gone into that pupae stage, and they're going to emerge as a whole lot more wasps to go out and get the rest of the caterpillars. And on the right hand, we've got a native bee. Again, just wonderful creatures that pollinate plants and do all sorts of cool things. That's actually a video that we're going to shit through that. And then moving on from the animals again to the this decomposition. Again, some of them are animals, small animals, but there's also all these other things that Ellen showed us, so I won't dwell on them. But yeah, fungi, protozoa, and these break down, hard to digest materials like lignified sort of woody plant materials and so on as well. So they will make use of the stuff that animals would probably reject, and still break it down and release the energy and the nutrients inside of it. They also, in this process of decomposition, they're also releasing all sorts of biochemicals that stimulate plant growth, stimulate microbial activity, and all that sort of stuff as well. So again, all of this is going towards helping plant growth. Just a quick picture of worms, and this is the red wrigglers. You can see they really reproduce quickly, and there's a few worm eggs in there, and they're living quite happily in that really lignified material. And between them and the other organisms in there, they're breaking it down. I've got a few videos too of these decomposers, but I'm just going to skip through them. Ellen already showed you an nematode and the saprophytic fungi, so we don't need to see them again. But I did just want to talk a little bit about, often when we talk about the decomposers, we think of them purely as aerobic organisms, but there are actually organisms that also live and break down materials that live in environments that are where there's less air. So like within the crumbs of the soil, or when it gets a bit wet, within the guts of animals. It's a very anaerobic environment, and they're actually an important player too. So it's not just about the aerobic organisms. So a lot of these here, this is a picture of some compost extract, or a video where I fed it with some milk and molasses. And you can see that there's a lot of activity in there. This is 18 hours later. There's still a lot of microbes that survived in this liquid where there's a lack of oxygen. And this is primarily the facultative anaerobes that have thrived in that environment. They can live where there's less oxygen. And they're an important organism because they're part of the the creation of humus. If we have aerobic decomposition, which involves lots of oxygen and burning off carbon, it's a fast way to break things down and mineralize things. But through more of these other pathways like anaerobic fermentation, the carbon is not lost so much from the system, and you end up with that humus in your soil, the carbon-rich humus, which is long-term stable humus. So it's really important. Whereas the aerobic pathways, the carbon turns over and gets released a lot quicker. So all of these organisms are important. And obviously, within a good soil, you're going to have crumbliness, this aggregation that's a result of all the activity by the microbes. And inside those crumbs, you're going to have these facultative anaerobes that can live with less oxygen. But then between the aggregates, there's going to be lots of oxygen where the proper aerobes can live. And obviously there's the gaps between the aggregates where water and air can move in and out. And within the aggregates, there's a whole rich environment that has conditions for certain nutrients to be released and to hold on to water and all that. So aggregation is a pretty important indicator of how healthy your soil is, because it means that if it's well aggregated, A, it had the biology to produce the aggregates in the first place, but then B, it's going to be functioning very well because there's going to be good air and water exchange and infiltration and things like that as well. And all those microbe groups will have a niche, a place to live in your soil. So summing up, the sort of the importance of these different groups, and I'm just going to spend a little bit of time on the slide because it's quite important. When we think of the symbionts that are living on the plants and living off exudates, they're living off high sugar food that's produced by the plants very not long ago. So they're pretty much, as soon as the plants are producing carbohydrates, they're put into the roots or they become available to these symbionts. And that's things like mycorrhizal fungi and nitrogen fixing bacteria, which then go out and get nutrients and bring them back to the plants and deliver them to the plants quite quickly. So there's a fast turnover of energy created, sugar energy used, provides a lot of energy for nutrient acquisition, which is then delivered straight back to the plants through the networks. However, when a plant grows into living materials that would then be eaten by an animal, it has to spend energy growing, and a lot of the organic material starts to be converted rather from carbohydrates into more complex carbohydrates like cellulose and stuff like that, which is harder to digest, takes more energy. And so energy gets wasted in the process. But nonetheless, digesters are pretty good at turning over the materials and releasing the manure and the urine back into the soil for the plants or the wastes. And then finally, you've got those really lignified materials like wood chips and straw and all the rest of it, which the decomposers take care of. And they're very hard to break down. A lot of the energy, the carbon and the nitrogen's already dissipated from them, as they were dying. And you need things like the fungi that Ellen was talking about, the saprotrophic fungi that, you see growing on logs and wood chips and things like that. Now, when people start a garden at home, for instance, often what they'll do to increase the fertility and get it going is bring in dead organic materials like compost or mulch. And so that's all fine and well, but we have to realize that we're dealing with the bottom part of the equation, right down the bottom here, where the turnover is slower, there's energy wasted, and it really might improve our soil, but not nearly as much as if we were growing living plants in our soil, and a diversity of plants that are putting out root exudates, that are feeding animals, and then finally dying and providing us with materials for decomposition as well. It's just a really important point that if we want to grow fertile soil, we need to do so with living plants. Otherwise, we're going to have to bring in lots of inputs, and it's really a slow path to Rome if you're doing it that way. So getting the party started, just keeping an eye on the time here. Obviously, the population and the behavior of the microbes is very much determined by the environment that we're in, and also our management practices, how we manage the soil. So the effectiveness of putting out biological products on your soil is somewhat limited, because if the conditions are not right, or they're not the right biology that's suited to that environment, or your management practices go and harm them, then putting out those materials putting out biology, microbes and things like that in your soil, they're just not going to survive. So we really have to look more at our management and our environment, and try and look at ways that we can kind of improve the situation that supports microbe populations. They're so complex, it's going to work well. And there are some slight exceptions to that, which I'll talk about in a second, but let's look at our management practices first. So one of the first things we can do is grow lots of different types of plants. Obviously, they're producing the carbohydrates, but if we want to support these symbionts that live on different plant species, we're going to need lots of different types of plants to support a wide range of different microbes that associate with the different plant species. So yeah, variety is really important, because the different plants host different types of microbes, which then promotes that diversity. And when you've got better diversity, like Ellen was saying, you've got a whole lot of services and resources that these microbes can get and perform, that then make your plants grow better. So diversity is obviously important. Plant diversity is where it starts. This is just a plot where I did a trial, a new garden, and I grew, a mix of different plant species together, but at the same time, I planted out plots of the species individually, and you can see where I planted the peas, same day, same soil, how well they grew by themselves compared to how well they grew in a mix, where there was this wider range of microbes there doing things and interacting and sharing, and it's just a richer and better network. This is just a photo of a similar thing where a grower in Kojonup had grown his usual crops, usual practices, but then he'd also grown a diverse cover crop in a field nearby, and he'd done it 2 seasons in a row. And the black soil is the transformation we got from those diverse cover crops, which is a really good indicator that carbon's happening. This is a video just of some water infiltration. I had a every hour watering. I'll let it talk for itself. Going to water it for a while because the water is just going straight in, and when it finally bubbles, I'll take it away so we can just see how quickly it's sucking in. So it's still not much of a puddle forming, even though I've been watering it for a long period of time. Try again. Just sucking straight in there. Might have to get a bit closer.
A little bit of lag on the video there, Mark, but still getting the picture, I think.
Go over here to where there's just normal soil. And do the same thing. That's the result, folks. Literally, that's been 2 meters away.
So, Mark, how long did you have the hose on there for? Your it was a bit hard to hear what you were saying there.
Yep. So yeah, I obviously had the hose on there for a fair while, just the whole length of that video, and it was obviously just going straight into the soil. And then I moved across to the side where there was, you know, where I hadn't done that, hadn't put that cover crop in previously, that previous season, and the water was just running off basically and taking some of the soil with it, and you could scrape away the soil at that point and it would just be bone dry underneath. So again, just having all those plants growing, a diversity of plants, all those root networks and microbial networks and crumbliness all helped. This next picture's a similar thing. I grew garlic dry because the soak was filled in where this garden was, and I had to rely on the soil to sort of hold on to that winter moisture right the way through to December when I harvested the garlic. And this was the day I harvested it, and you could still see that moisture and the fungi in there and all that, because the soil had become functional from all the work we'd done previously with diverse plants and the microbial networks. I just put a list in there of cover crop species that I would use in a mix, like an autumn mix like that. And I can share these resources with you guys as well. But yeah, you're just looking at a range of things that grow fast and well and feed the biology. So these microbes that live on plants, it's a pretty important point that when you're making compost, you're not breeding these microbes because they live on living plants. So compost is just a primarily dead material. And as much as that's important, if you want to breed symbionts, you have to let plants actually complete their life cycle, because when annual plants are dying, sorry, I'm just going to close the door. My son talking next door. When the plants are growing and when they go into reproduction and start to die, the microbes that live on them are also triggered, or it's their cue to also reproduce, and then they release spores and eggs and stuff that in large numbers that are able to survive over the dormant season. So if we want our symbionts to be able to breed and survive over summer, we need to let a diversity of plants finish the annual plants finish their life cycle. The perennial plants are okay because they keep living through the season, but these ones die, and without that support of a living plant, these organisms have to be able to reproduce and go dormant. So this is just a field where we've grown a cover crop, and you can see we're just letting it go to seed. And the following, I'm just going to come back to this slide. The following year, that's what it looked like. So this was 6 weeks after the break, and because there was so much seed that had set seed, but also the soil and all those microbes, when these plants started growing the following season, they really jumped out of the ground. And you can see, you know, they already had their microbial associates in the seed and in the soil from when they grew previously, and we'd let them go to seed. This is a recipe of how you can go about just growing, you know, these cover crops and then shaking off the soil from around the roots to create, you know, like a culture of these that you can then use to mix with potting mix or coat your seeds with. So this is different to coating your seeds with compost, which is not the symbionts, it's the decomposers when you're dealing with compost. So yeah, anyway, I'll share that recipe. Basically, you just grow a cover crop and let it go to seed and mature, and then you harvest the soil around the roots and the root fragments. And then there's composting. I love the Johnson Sioux type compost because you don't have to turn it. It's not a hot compost, so it doesn't cook the fungi or anything like that. It stays pretty aerated and cool because it's done with pipes and things like that. If you haven't come across that system, it's a great system to look up. And it hopefully produces a compost that's really rich in humus, which is putty-like this, and high in fungi because you haven't disturbed it too much. And a lot more of those facultative anaerobes because it's not an aerobic compost that you turn all the time. It's static, and so even though it has airflow, it does kind of end up looking a bit like this, where there's a lot of aerobic and anaerobic environment within it for different microgroups to survive. I was saying that it's hard to add biology to your soil and expect it to do too well because it just might not survive out there. But one thing you can do is put biology around the seeds of your plants, because when your plant first germinates, and especially dormant biology, like we talked about where we let the plants go to seed, or when you let your compost really mature, and a lot of the biology in there is much more dormant, they can survive better in the environment. But by putting them around a seed, it means that when the plant germinates, there's a food source there for those microbes, and the microbes that live off those types of plants will survive. Around the roots of the plants, there's much less diversity. The microbes living on and around the roots of the plants are specialized that associate with that type of plant, but there is a lower diversity but high amount of activity because the plants are putting out specific root exudates for those microbes. Whereas in the bulk soil, we've got a lot of different microbes, a high diversity, but they're mostly dormant because there's not living plants going on there. So yeah, there's just this sort of contrasting environments within the soil. And if you want to add inoculants and things to your soil, that's probably going to be your best bet in terms of adding biology that's going to survive. You pick specific inoculants or broad spectrum inoculants that survive on the plants, and you add them to the seed. So this is an example of that, where we're just coating some seed, in this case with a bit of compost slurry. We've made some, you can see it in the background, the brown, we've mixed compost with water and we've coated the seed. And most seeds in a healthy ecosystem will be germinating in that rich layer beneath the organic matter, where it's kind of like compost anyway. So they're used to germinating in that environment. This is just some potatoes that I coated in the slurry. And again, as they get going and they got that healthy start, you can see all that soil clinging to the roots and off they go. As a general trial, or just a way of just testing the effectiveness of these different treatments, I will get the soil from the field that I'm going to plant and the crop that I'm going to plant, and I'll try different things like using some soil that was shaken off, dead plant roots to keep the symbionts in there, or I'll get some compost extract and I'll coat the seeds in that. And in this case, I coated these peas on the left-hand side with some compost slurry. And again, I'll provide the recipes for that. And then the ones on the right didn't get it. And you can see just that small treatment has triggered a huge difference in the growth in this particular environment in this soil. So that's a treatment that would be worthwhile doing for us as farmers if we were planting peas in that environment. Similar thing here. The ones on the left got 2 treatments of compost extract, just watered in with a watering can. And as things dried out, they just held their form and color a lot better. When I make potting mix, my simple recipe that's very effective is just using good, healthy garden soil, or particularly soil from around dead cover crops that you've let go to seed again, and then mixing it with compost. So you've got the decomposing microbes in there and all of the chemicals they produce, and you've also got the symbionts in there as well. And it just really makes for a healthy mix. Brassicas always show a deficiency, or if they're not doing, if the potting mix is not good, they show it, and these brassicas are thriving. And this is about 4 weeks after planting, or maybe longer. You can also do bioferments. Probably haven't got time to get into them today, but there's lots of resources out there on how to make products with these things, these fermenting organisms, same thing that we'd use to brew beer or make sauerkraut. And they also exist in our soil. They're the facultative anaerobes, and they're basically producing these sort of acidic organic acids similar to what you'd get if you bought some fermented fish or some fermented seaweed, and they help with various things like making nutrients available. They chelate nutrients so that they become stable and plant available. So you can use, you know, these different biostimulants that you buy on the market, or you can make them yourself with something like a bioferment, and you can mix them with nutrients and so on to just help with general uptake and so on. Just to finish off with some of our other practices like fertilization, you know, being aware that high salt fertilizers do burn biology. They haven't got thick skins like we have, and so they burn. And just looking at your fertilizer sources and how you put them out. And again, this is where mixing some of these fertilizers with biostimulants like humic substances and stuff like that can buffer them, make them softer, and also just, yeah, improve their form and availability to plants. So some fertilizers are saltier than others, and that's just a chart just showing where, you know, if we use something like calcium sulfate, like gypsum, it's really not that burny, even potassium sulfate. But then if we get up to some of these other things like ammonium nitrate or sodium nitrate, they really, really burn the biology, and you've got to be careful how you use those. There's a range of things that we can do to be, you know, careful with our fertilizers. So, you know, just again, not putting too much soluble fertilizer near the seeds and the germinating roots when you want your plants to get off to a strong start. Doing smaller applications more regularly, using these humic substances to buffer them, and combining them with all these, you know, these biological foods. And just remembering too that fertilization can be beneficial because it can help plants grow. So, you know, I'm not saying fertilization is bad by any stretch of the imagination. I'm just saying it's being aware of how to do it without harming the microbes too much. Another thing that's really important with our nutrition is foliar nutrition, which doesn't hurt microbes because it's put on the leaves. But particularly when it comes to trace elements, when we put the trace elements on the soil, and you can see when we look at zinc, iron, copper, manganese, only about 1 to 2% of them ends up in the plants. So it's pretty useless applying trace elements to the soil in some ways. There are some exceptions, but as a foliar, you can get them straight into the leaf. And so, you know, trace elements like iron and manganese are really important for photosynthesis, which is carbohydrate production. So you really want to try and get those things into your plants. So something like foliar nutrition can be a big thing too. And then the way we treat our soil in terms of disturbance. This is just a video from a friend of mine in South Australia, Tom Robinson, and he's using, you know, a stripper front to harvest. So he's leaving lots of residue on his soil, and he's using these disc seeders that, hopefully this works.
If it won't play, Mark, we might have to leave it there, just so we've got enough time for questions.
Yeah, this is the last one here. If that's okay, or we can stop.
No, that's fine. It's just not, the videos aren't playing.
Yeah.
There's a little bit of lag there, that's all.
Yeah. Okay, so yeah, it was just a machine that, you know, again, it's leaving all that residue and it's not really disturbing the soil, and it's just cutting into it and planting the seeds. So, you know, just really not upsetting the microbe habitat by going and digging it and exposing everything to the sun and the air and the, you know, the wind and the rain and so on. This was just a photo. I took a couple of photos of a farm that I live on, and it's just basically, it was a pasture, and they built a dam and they scraped the topsoil off the dam and put it in a big pile. So this was the best soil they put in a pile, and then they left it there for a couple of years. And in those couple of years, obviously it didn't have plants growing in it. It didn't have pasture growing on it anymore. And so all those biological processes were no longer being supported. The symbionts couldn't survive because there were no plants growing. The organic matter was not being replaced because there was no litter getting put on it. And basically, in essence, the topsoil died and became just mineral matter. So even though it had all those nutrients that were in there still, the biology had been sort of essentially shut down. Then we went and spread it a couple of years later over the paddock, and we seeded a cover crop over the whole farm at the same time. And these two photos were taken, one at the top left-hand side was where the pasture, the paddock had just been in pasture for the last, you know, for as long as I can remember. So we hadn't disturbed it in that way. And then the picture on the bottom right was the damn topsoil that had been spread. And because it had died after being in a pile for so long, the growth response we got was absolutely terrible compared to the other picture, which was just 50 meters away. So it just goes to show that, this maintaining your biology is an ongoing process. You don't just fix your soil and add stuff and then it's done. You have to keep growing diverse plants in there, making sure that you've got all those microbes being supported. Also, the red-legged earth mites really hammered the stuff in the bottom picture because it just didn't have that biological immunity as well. So it was a combination of poor growth, poor immunity, and various other things as well, probably soil structure and things like that. So yeah, I guess just rounding it off, that importance of just continually keeping your biological systems healthy. That's me.
Thank you so much, Mark. So much knowledge there, and thank you for sharing. And I would love to see those soil recipes, as I'm sure that all of us would. I'm going to throw out to the room now and see, does anyone have any burning questions that they would like to ask? Ella jumped in straight away. That was like crazy quick.
That was me clapping, not putting my hand up.
I got my emojis mixed up, sorry.
Go on, Casey, you're going to be young.
Does anyone have any questions yes, Amy?
Thanks, Mark. That was amazing. And to the other speakers too. I wondered, you mentioned about that, the topsoil dying and going back to mineral. And I wondered, is there any way, or what's the process and how long does it take to recover soil like that? Do you just sort of have to go back to square one and start doing all of those steps that you'd gone through? I'm just thinking about my veggie garden at home that got left to go to rack and ruin, and wanting to revive it.
I mean, the fastest step is obviously through that living root exudate pathway. So getting a diversity of plants, and there's particularly plants that grow fast and produce lots of root exudates, like that list that I had of cereals and legumes and oil seeds and things like that. And yeah, you could even help them a bit by maybe getting some compost in there to help them grow, or coating the seeds with compost. And then finally, if you want, when that's finished growing, you could feed them to the chickens, like animals, feed the litter to the animals and let them break it down a bit and digest it a bit, and then put that back on the garden as well. So you're kind of using all the different groups, but of those, the most important thing is getting diversity of plants in there, growing actively and putting out root exudates and producing the carbohydrates that feed everything. Yeah.
Thank you.
Can I just add? Oh, sorry, go on, Amy.
Oh, I just had a follow-up question.
So just to add, just as another comment, is that our soil virtually never ever completely dies from the microbial, the content. It actually tends to go into a dormancy. So even in regular soil, about 80% of the microbes in the soil at any one time will be dormant. The other 20% will be active. And so provided we haven't had some, like a blowtorch go over it or something like that, the microbes, when they're just slowly going into this poor situation, will go dormant. And so, like Mark's saying, it's about activating them. And one of the best ways of activating them is those exudates from living plants.
Fantastic.
Great answers, guys. Thank you. I'm just learning so much today. Any other questions? You've left everyone stunned.
I actually do
follow-up
have one now. That's
question? Ella.
not just clapping my hands. So something about what. Well, thank you, firstly, Ellen and Mark. That was awesome. It was so interesting. I think about sort of that soil microbiology in a revegetation context. I work out in broadacre agricultural areas, so we're talking, annual crops, a lot of annual pastures, and then we're coming in often revegetating into paddocks that have been in this cycle for a very, very long time. And of course, one of the big things we do is weed control. So we come in the autumn beforehand, we spray everything out, we kill the weeds, and then we put our little plants in. And obviously that seems to be complete juxtaposition to what we're saying about the importance of of those live plants in there. So what's your advice in how do we manage that? Obviously, if we can improve the soil health, we can have that more of that microbiology up and running before we put our little seedlings in, that's better. But coming from that background and knowing that weed competition is such a big problem, especially out here, it's dry. How do you balance that? You know, moonscape versus everything.
Yeah, it's a tricky one. I mean, yeah, it's all great to have plant biodiversity, but obviously weeds are going to, you know, compete and you know, take moisture out of the soil and all of those sort of things. So your weed control is pretty paramount for establishing those native species. However, wherever you can, potting up, you know, your native species in really healthy potting mix in the first place that's got a mix of, like, you know, good symbionts and maybe some compost as well. All of that will really help them to have a strong microbiome when you plant them. And, you know, it certainly wouldn't hurt to water them in with, you know, or use inoculants where you can, you know, with the different microbes, just to give them that support when they get going. But yeah, you obviously, you know, you do have to take care of weeds. And like Ellen says, you know, using a herbicide will kill or will hurt certain microbes, but there's a lot of them still there, dormant, ready to go. And yeah.
Yeah. And so to that, I'd add that if you can restrict the spraying to perhaps even strips, if you can. So it's obviously, it's like a Goldilocks principle of things, nothing at extremes, and just trying to find what's right for your situation. One of the parts might be that you can actually make up your compost that you might inoculate the seeds with, including a little bit of soil from some healthy native vegetation that's close by, so that you've got those indigenous microbes in there that are going to already be saying, oh, this is a pretty good place to live. So that's the one part. And the other part to remember is that many of the herbicides and glyphosate, a number of them actually chelate minerals. And so that means that they lock up minerals that are in the soil. And so it may be worthwhile actually looking at a mineral assay for the soil that you're looking at as well for availables. And I mean, I know that you're talking about native plants, but they obviously still have requirements. And so that might be something also just worth considering, Ella. It's in amongst those things. The water, and so the biological, using like a biological mix and sort of that watering in, I believe is just really valuable, because if you can create that soil structure around the roots as you're growing, then that's actually going to set it up like one of those spirals, an upward positive spiral for things to happen.
I think the challenge in that, in what you're both saying, because that makes perfect sense, is scale. You know, what you can do in a backyard veggie patch sort of thing, you know, versus I think we've got 120,000 seedlings going out across 17 sites next year.
Yeah. So when we're talking about
just
compost,
sitting here inoculating every little.
oh
my God, we can't do that.
So that could be challenging. It could be a possibly. This is just a possibility. So remembering that I'm not talking so much about making the compost as the compost extract, and the compost extract. So where you're effectively, basically, like I showed you with the test tube, so you're basically washing the microbes off the soil particles. That will go that will go a long way. And so even if you perhaps had some sort of a dunking station where you could dunk the things in, it may just contribute. And I mean, I think from what Mark showed, that just doing some of those little things provided quite an improvement. And that diversity of the seedlings, when you're planting the seedlings, that there is that diversity of families so that you can get those interactions.
Yeah, the potting mix, the planting treatments is always the logical place to start, because you can just add stuff to your potting mix or water your seedlings with a compost extract and inoculants and things like that. So that's sort of, you can do that all in the nursery, and then once you get it out in the field, obviously it's impractical to do too much, and most plantings just get done and left, and hope survival of the fittest from there on in.
Yeah, that's not our nursery either, so we have no control at that point.
Yeah.
But yeah. Okay, thank you. I'll let Bill jump in.
Yeah, just some experience. That I know it's labour intensive, but even a bit of mulching around the little seedlings one, it adds nutrients and food for the microbes, but it also provides a lot of protection and controls some of the weeds in that immediate zone around the plants. But it's labor intensive. But if you do it at the same time, you've got a bucket of mulch and you put your plant in and scatter some mulch around, that seems to work really well.
Yeah, that's a really important point, Bill, because like in our Mediterranean environment, we don't have many living plants growing, annual plants growing through the summer. And having that mulch layer is the habitat and the protection and the food source for our decomposing organisms. So yeah, it's really important. So, you know, like in an ideal world, you'd actually you'd spray out your weeds, seed a cover crop, let it go to seed, let it be mulch, and then plant your natives into that.
Yeah.
But yeah, obviously it's just time and money. But yeah, that mulch is really important in the summer here. Really important. Yeah.
Okay, everyone, I jumped in there because Katie had to disappear momentarily to pick up Coco. So lovely to see you all. I'm Jack, the Executive Officer for WALMS. Thank you for what looks to have been a great conversation. I got a quick text from Katie saying it's been terrific. I think something just went into the chat. Let's have a quick look. Was that you, Amy? No. Oh, Amy from Wheatbelt NRM. You could water the seed trays with inoculant before they're planted out. Yes, Ellen says, great idea. So some good practice tips there. Anything else from the floor? Anything else to be asked of our speakers, Amy?
No, just to say, fantastic presentation from both of you. I'm very impressed. Really enjoyed it. Thank you so much.
Thanks, Bill. Over to you, Amy.
Thanks, Jack. Sorry, my question was similar to Ella's actually. So I used to work for Greening Australia and we did large scale tree planting, but with direct seeding. And I think, yeah, I really liked your idea about sowing that green cover crop and letting it go to seed. But I'm wondering some of those seed treatments and dunking things that you talked about. Have you got any experience with some large scale sort of direct seeding? Some of the sites that we used to get were pretty horrendous in terms of the soil quality. And so, yeah, aside from the green cover crop, is there something else that you could recommend for large scale re-veg?
Yeah, I mean, there might be other things, but one thing that just comes to mind straight away, obviously, and you'd be aware of this already, is that the quality of the seed that you use is huge. Like, and you know, because the seed has within it not only the energy source and the initial nutrients, but it also has a microbiome itself. So the plants actually deposit a lot of their symbionts in the seed when they set seed, and then those are there so that when it germinates, it can get off to a good start. So, you know, sometimes using fungicides on seeds and things like that's kind of counterintuitive. But if you can collect seed from an environment that's really healthy and the plants are healthy, then they're obviously going to get off to a much better start. I think other than that, you know, just getting things like if you have soil from a healthy environment in that area that has already those microbes that live in that area, and you go and collect soil from, you know, the topsoil and you sieve it, and you basically get a fine mix that you can then actually toss with the seeds or like lightly moisten it and let it slightly coat the seeds. And then that again puts those microbes from the healthy environment into the unhealthy environment. And if you do it at the right time of year, they should be mostly dormant, so they survive until it rains, and then the seed germinates and off they go. So yeah, that's just an idea. And then of course there's also compost options as well, where you get compost extract and do a similar thing. You mix the seeds with compost extract or something like that, and that also helps. So all of those things, if you can find a system or a strategy to do it that's, you know, not too labor intensive, and I think, yeah, most of those things can be done fairly easily once you've got a bit of a system in place.
Yeah, awesome. Thanks, Mark, and thanks, Ellen, too.
It's probably not as helpful, Amy, but something Dr. Christine Jones talks about is that research that shows there's what's called a legacy effect. So we talked about how each plant family has its own set of functional microbes. And so what's been found is that if you plant a plant in the soil, it actually releases some of those microbes that Mark's been talking about into the soil. Some of them, not all of them, but some of them will remain there. And so that if you come back and you're planting similar plants, next year, like, or the replacement ones, they're actually also going to be already having some connections in there. So it's basically like those microbes are going to know somebody that knows somebody that knows where the grocery store is, type of thing. So
Yeah, cool.
I know it's not much help in the beginning, but maybe it's helpful later on.
Yeah. No, that's great. Thank you. Thank you both.
Okay, look, beautiful timing, 3 minutes to 3. Any last words? Anything left in the room? Anyone wants to ask, or we'll leave it there? Case will send out a bit of a wrap up. She'll send a link to the recording once that's been fully processed. And I think she sends out a little survey too. We like to get feedback on our Landcare Build Skills so we can add that into the mix for what to do next to bring you further value. Soils, I think, is going to be a big conversation forever for us to give access to people to deepen their knowledge and understanding of, particularly as the climate continues to do what it does in terms of drying, particularly here in the southwest, and what impact and what changes that causes. I think it's really a continually moving conversation, isn't it, that we need to keep having. Fantastic. Thank you, Ellen. Thank you, Mark. Thank you, everyone, for making the time today to come and join us. I'm glad you enjoyed it. And I think our next catch-up is the Landcare Checks In, which is about how to use the Atlas of Living Australia to best effect. So you'll hear some more about that in the next couple of weeks. If you're coming to the annual gathering on Friday, love to see you there. Thanks,
Thank you.
everyone.
Thank you.
Bye for now.
Bye.